here is just a quick little look at the different things that have been considered for Middle East respiratory syndrome. Now, this is another coronavirus. It doesn't spread quite as well from person to person, but it is much more dangerous. Current case fatality rate is something like 30%. So 1 out of 3 people that get this virus die from this virus at the moment. And so, yeah, it's another case where a vaccine is needed and a vaccine is not yet available, but here are the candidates. And this is nice because it puts them all in one spot. So this is the paper that I'm pulling from. You can see it's pretty recent. And this is the best. And one of the papers out of this is the one that we're going to look at because it's similar to the other one of the Merck vaccines. So skip past that figure, which is a really weird looking picture of the spike protein. And here we go. This is what we want to look at. Table 1. So this is fantastic. This is like all of the MERS vaccine effort in one little spot. And so let's go. Let's have a look at this. All right, so what do we have? What's been tried, what's been done, and how far has it worked? So for MERS coronavirus, there are people that have tried to just make, so subunit means you're making a piece of the protein, putting it into the person, and then seeing whether the person makes enough of an immune response that they would be protected from the actual virus. So here they're putting in just the receptor binding domain of the MERS spike protein. Now, MERS virus binds to a protein called DPP4, but it still has that same shape of receptor binding domain, and it still is either up or down. It's the spike protein works the same way, it just sticks to a different thing. So this is still the same part of the protein as the receptor binding domain of SARS coronavirus 2, basically. And so the M and the P here mean, so humoral response, actually let's do the titles first. So vaccine type, great. Humoral response means antibodies. Did we get good antibodies? Cell-mediated response means did we get good T cells, for example. Protective means did we get enough of an immune response that we could put in the virus and the animals would not either get sick or die, depending on what the model is. Clinical trial is actual human use, and as you can see here, the farthest we've got anyway, is phase 1. And phase 1 is just safety, and it's usually pretty small. You can have like 20 people in your phase 1 and it'll still count. And this is just normal, healthy volunteers, and you're seeing whether or not they get sick from the vaccine that you're giving them. Phase 2 is similar to phase 1. You're checking that they don't get sick, but you're playing around with the dose. And what you're looking for is, does it look like these people are probably going to be protected? So do they make antibodies? Do they make T cells? Stuff like that. And then phase 3 is a proper protection against challenge. So do you get less disease from the actual disease if you've had the vaccine? Or do you survive the actual disease more than a person that doesn't have the actual vaccine? So yeah, that's the phases, and yeah, we're in a real early stage with all these. The numbers over here point to the papers, and yeah, it'll pop up, and from the title you can kind of get exactly so here the receptor binding domain is 231 amino acids, and yeah, it elicits, which means it makes the in this case mouse or so the M is for mouse, the P is for a non-human primate. So probably a macaque monkey, but there are different ones that mimic different parts of the disease. None of them is a perfect match for the actual human disease. And let's see, the C I thought might be camel. There we go, yeah. So MERS is a disease that is given from a bat to a camel to a person, and so they're trying to protect camels from this, and they're trying to protect people from this ultimately. But if you protect the camels, then you've also protected the people. So yeah, that's a good thing to be able to do. Okay, so here are just injecting some version of the protein, so just the receptor binding domain, or the entire spike stuck to some kind of carrier. And we could look in this paper and figure out what they stuck it to, but nanoparticle just means some small lump. It can be a little styrofoam particle, it can be a little chunk of gold, it can be a lot of different things, and whatever. They're all nanoparticles because they're some number of nanometers in size, I think. Less than 100 nanometers normally, sort of between 10 and 100. Okay, so this one they're just putting in the spike protein. This one they're just putting in the N-terminal domain, which means S1 part of spike protein, which is weird because S1 doesn't properly fold up without S2 being there. So this is going to be a kind of a denatured version of the protein. But yeah, you can see that these things have different amounts of protection. So you've got good antibody responses for all of them. And you've got, in some cases, they've gone in and actually looked for T-cell responses, so that's good. And yeah, what you want to see is that they are protective. They'll protect an animal. And so, yeah, most of these protect at least something. And it would be better if it protected primates, but not all of these have even been tried in primates. I think that's the problem. Okay, so we got your protein version. So inject a protein, wait for a response, and then see if the response protects you against the virus. We've got DNA, which codes for the gene. And you see which gene they're putting in? S, S, and S1 or S. Yeah, these are all based on the spike gene. So this idea that SARS coronavirus 2, that all these vaccines are based on the spike gene, this is not a new thing. People have been making this same, I don't know if it's a mistake, but this same attempt to sort of simplify the immune response down to an anti-spike immune response. They've been doing this, a long time, and in a lot of cases. This is PVAX1. This is a DNA vaccine that they've actually got all the way to phase 1. Now, DNA vaccines are things that are known to work very well in mice, and they usually don't work particularly well when you get them to humans. There are some primates where you can get them to work, and this one has. So it could work, but it would be the first one that has worked properly in humans. So, yeah, I don't know. Eventually there has to be one, right? Viral vectors. So these VEEVs are a thing called an alpha virus, just a different kind of virus, and we're using it with the spike protein or with the nucleoprotein over here as a way to make these proteins from MERS coronavirus inside the body. And you can see this one with the nucleoprotein is actually protective against MERS virus challenge in mice. So that's kind of neat. MVA is a vaccinia virus, so this is the vaccine for smallpox, and they've just crammed an S gene in there from MERS coronavirus. So this would probably protect you against both smallpox and MERS virus at the same time, if it worked, and it seems to work in mice and camels, so good for them, and it's in phase 1. These ads, this is an adenovirus shell, and adenovirus 5 is one that goes in very well in humans. Adenovirus 41, different one. CHAD is a chimpanzee adenovirus, and there's an Oxford 1 and an Oxford 2 strain. And you can see they're just plugging the spike protein into all of these things. Newcastle disease virus is a bird virus, but they're using it as a way to deliver spike protein. VSV, this is just like we were talking about with the spike protein. And this one actually works in primate models, so that's neat, but we aren't into clinical trials yet. And we've got a rabies virus, S1. Oh, and the VSVS was not tested in a... they didn't look for protection, whether it actually blocked the animals from getting disease later on. So they're not that far along yet. But the rabies-based S1, so just the top of the spike, jammed into a rabies virus, that one does actually protect mice against challenge. And that's the paper we're going to be covering. But over here we've got baculoviruses, which are normally an insect virus. This one's making S, E, and M protein, so that's pretty cool. That's a little bit different. This one's just making the receptor binding domain of S. And then down at the bottom, you've got whole virus. So this is you grow up a great big pile of MERS coronavirus. You kill it with formalin, which is formaldehyde solution. Or you can take away the E protein, and then the virus does not grow well. Or you can take away this one, non-structural protein number 16, which is a protein the virus uses to hide. And so the virus is shut down pretty quickly in that case by the immune system. Or you can take out open reading frames 3, 4, and 5, which is really severe. And that's another way. You just, carve chunks out of the virus as a way to attenuate it. Now, with these deletions, it looks as though they could actually recombine with the actual virus, and then boom, you'd be back to normal again. So then, I don't know, they're pretty safe, but oh, and these are yeah, these are whole virus. This is killed virus. These guys would all be live MERS coronavirus, but missing big chunks. So there'd be some safety concerns there, but they're missing pretty substantial chunks. So anyway, there you go. That's where we are. A lot of ideas, a lot of things that could eventually work out, but nothing that's farther along than phase 1 as of fairly late last year, and I don't believe anything is even into phase 2 yet. So if we get a coronavirus vaccine of some sort, the first one is probably going to be for SARS coronavirus 2. And it's probably going to be one of the approaches that we are covering, because these are the leading ones. So you are very much dialed into exactly what's going on right now. And that's kind of neat. Most courses can't be quite this, I don't know, finger on the pulse, contemporary, switched on. But hey, right time, right place, right class. There you go. Thanks very much. This has been, an overview of vaccines.